Welcome everyone. In today's lecture, I will be discussing about the physiology of vision. Before I start, I am glad to mention that this presentation is a part of initiative taken up by the Zoological Society of Assam to reach out to the students during this COVID pandemic, especially those who were pursuing graduation in zoology. I have dedicated this lecture to my honored teacher, Professor Jogin Chandra Kalita, who is the professor in charge of Animal Physiology and Biochemistry Laboratory, Department of Zoology, Guwahati University, Assam. He has taught me to have a vision in the field of zoology and encouraged to fly high. Here I am happy to mention that he also glorifies the position of General Secretary of this forum. Now let us begin with today's lecture, the physiology of vision. How we can see the beautiful world, the mechanism behind it. From the presentation, we can acquire knowledge in various important perspectives. In structural perspective, we can learn the anatomy of the eye, which is often compared to a camera for the formation of image, and the retina, which is serving as film. However, the structural elements of retina is far more sophisticated than the most expensive camera. In the retina, the eye has a layer of photoreceptors that respond to the light. Great work has been done on the physiological perspective of vision. In fact, it is said to be the most studied and perhaps the best understood sensory system. The way the components of the visual system operate to set up conscious visual images is very interesting. Here, phototransduction, that is, conversion of light into the electrical signal in rods and cones, play an important role in color detection. The signal goes through the neural circuitry of retina, then leaves the retina via the optic nerve. The visual nerve signal then goes to the visual cortex of brain, which comes under the heading of central neural physiology of vision. In the clinical perspective, we will learn the common diseases of visual dysfunction. Lastly, and very interestingly, we will have an understanding of cutting-edge research on vision in research perspective. We introduced, vision is the most important spatial senses in human. We depend on vision for most of the human communication. Interestingly, 70% of all the sensory receptors of our body are present in the eyes. Now, what we see or our visual system detects. The visual system detects and interprets the photic stimuli. In vertebrates, the effective photic stimuli are electromagnetic waves between 400 and 700 nanometer long. Such wavelengths constitute the visible light. Basically, to see, perceive and recognize anything or an object. Its eye has a lens system that focuses the light, a layer of photoreceptors that respond to the light, and system of nerves that conducts impulses from the receptors to the brain. Now, to figure it out how all this work, let's start with the eye anatomy. The wall of the eye is formed of three layers. The outer protective layer of eyeball is the sclera, or the white of the eye through which no light can pass. It is modified anteriorly to form the transparent cornea through which light rays enter the eye. The lateral margin of the cornea is connecting with the conjunctiva, a clear mucous membrane that covers the sclera. Just inside the sclera is the choroid, which is vascular layer that provides oxygen and nutrients to the structures in the eye. Innermost is the retina, the neural tissue containing the photoreceptors. Apart from these layers, the eye also contains a lens to focus the light on the retina and fluids called the aqueous and vitreous humor. The light enters the eye through the cornea and passes through the aqueous and vitreous humor and then focuses on the retina. Light rays are bent when they pass from a medium of one density into a medium of different density, except when they strike perpendicular to the interface. The bending of light rays is called refraction, and it is the mechanism that allows one to focus an accurate image onto the retina. The refraction takes place in four interfaces: the interface between air and the anterior surface of the cornea, the interface between posterior surface of the cornea and aqueous humor, the interface between aqueous humor and anterior surface of the lens, and the interface between the posterior surface of lens and the vitreous humor. The refractive index, which is the ratio between the speed of the light in vacuum and in medium, in case of air is 1, while the refractive index of cornea is 1.38, aqueous humor is 1.33, lens is 1.40 and vitreous humor is 1.37. If all the refractive surfaces of the eye are algebraically added together and then considered to be one single lens, the optics of the normal eye may be simplified and represented schematically as reduced eye. This is useful in simple calculations. In a reduced eye, a single refractive surface is considered to exist with a total refractive power of 59 diopters when the lens is accommodated for distant vision. As we know, the lens of the eye can focus on image on retina. The image is inverted with respect to the object. Very interestingly, the mind perceives objects in the upright position despite the upside down orientation on the retina because the brain is trained to consider an inverted image as the normal. The image which is formed on retina has millions of photoreceptors which convert light energy into the electrical signals that brain will detect. Before going to the photoreceptors, retina itself has two layers: the outer pigmented layer and inner neural layer. This figure shows the functional components of the retina, which are arranged in layers from the outside to the inside. These are pigmented layer, outer nuclear layer containing rods and cones, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, layer of optic nerves or stratum opticum. The optic nerve carries information through the thalamus to the visual cortex. This is the sequence of absorption of light and processing of human vision. Thus, we got to know that retina is the light sensitive portion of eye that contains the rods, which are responsible for black and white vision, the cones, which are responsible for color vision. Each rod and cone is divided into an outer segment, an inner segment, and a synaptic terminal zone. The outer segments are modified cilia comprised of regular stacks of flattened sacules or membranous discs. The inner segments are rich in mitochondria. This is a region that synthesizes the photosensitive compounds. The inner and outer segments are connected by a ciliary stalk through which photosensitive compounds travel from the inner segment to the outer segment of the rods and cones. Photosensitive compounds that react to the light to initiate action potentials in the visual pathways. How action potential has been initiated? To answer this, let us move towards the basis of photoreceptor potentials. So light is the key factor to see. And what happens in light? The closure of the sodium channels. In dark, sodium channels in the outer segment are held open. Light enters, some of the sodium channels close. This produces hyperpolarization of the synaptic terminal of the photoreceptor. This in turn reduces the release of synaptic transmitter. This generates action potential that are transmitted to the brain. Here I have presented the photochemistry of rhodopsin, and the same principle can be applied to the cone pigments. Now the sequence of events in the photoreceptors by which incident lights leads to the production of a signal in the next succeeding neural unit in the retina is summarized here. The phenomenon is called phototransduction, by which light energy is absorbed by photopigments and converted into a biochemical signal that leads to the hyperpolarization of the photoreceptor. When there is incidence of light, structural changes happen in the retinal pigment. Here I am talking about rhodopsin. Rhodopsin is comprised of retinal, an aldehyde of vitamin A, and a protein called opsin. In the dark, the retinal is present in the 11-cis configuration. Light changes the shape of the retinal, converting it to the all-trans isomer. It separates from the opsin. This in turn alters the configuration of opsin, and the change in the opsin activates its associated heterotrimeric G protein, which in this case is called transduction. The activated transduction activates phosphodiesterase. Activated phosphodiesterase hydrolyzes cyclic guanosine monophosphate, that is CGMP2 5-guanosine monophosphate. Cyclic guanosine monophosphate normally acts directly on sodium channels to maintain them in the open position. So, the decline in the cytoplasmic cyclic guanosine monophosphate concentration causes some sodium channels to close. This produces the hyperpolarizing potential. The hyperpolarization reduces the release of synaptic transmitter glutamate, and this generates a signal in the bipolar cells that ultimately leads to the action potential in the ganglion cells. The action potentials are transmitted to the brain. Now, the brain turns the signal into the images. That what we can see. The human eye can detect almost all the gradation of colors when only blue, green and red monochromatic lights are appropriately mixed in different combinations. This theory of tricolor mechanism of color detection was proposed by Young Helmholtz, and they had postulated the existence of three kinds of cones, each containing a different photopigments and are maximally sensitive to one of the three primary colors, blue, green, and red. The blue sensitive, green sensitive, and red sensitive pigments containing cones show the peak absorbance at light wavelengths of 445, 535, and 570 nanometers respectively. Here I would like to draw the attention towards the sensation of white or any spectral color, just a yellow, can be produced by the mixing of blue, green, and red light in the various proportions. Therefore, these three colors are called primary colors. Let's see how nervous system can interpret the colors. We can take an example of a color from our visible spectrum, let's say orange. An orange monochromatic light with a wavelength of 580 nanometers stimulates the red cones to a stimulus value of about 99. It stimulates the green cone to a stimulus value of about 42, but the cones not at all. Thus, the ratios of stimulation of the three types of cones in this instance are 99:42:0. The nervous system interprets this set of ratios as the sensation of orange. Conversely, a monochromatic blue light with the wavelength of 450 nanometers stimulates the red cones to a stimulus value of 0, the green cones to a value of 0, and the blue cones to a value of 97. This set of ratios, that is 0:97, is interpreted by the nervous system as blue. So, while white is the combination of all the wavelengths of the spectrum. Now, coming back to the previous concept of phototransduction, where light energy is converted to biochemical signals in retina by neural circuitry that include the photoreceptors, the rods and the cones transmit signals to the outer plexiform layer, where the synapse with bipolar cells and horizontal cells. The horizontal cells which transmit signals horizontally in the outer plexiform layer from the rods and cones to the bipolar cells. The bipolar cells which transmit signals vertically from the rods, cones, and horizontal cells to the inner plexiform layer, where the synapse, the ganglionic cells, and amacrine cells. The amacrine cell which transmits signals from the bipolar cell to the ganglion cells. The ganglion cell which transmit output signals from the retina through the optic nerve into the brain. Thus, the visual message about the world around us is carried by the two optic nerves via the thalamic lateral geniculate body to the visual cortex of the central nervous system along with the specific pathways. The fibers from its nasal hemiretina decussate in the optic chiasm. In the lateral geniculate body, the fibers from the nasal half of one retina and the temporal half of other synapse with the geniculocalculin tract. The tract passes to the occipital lobe of the cerebral cortex. The processing of the message at different levels results in conscious perception of stimuli as well as the reflex responses to them. The whole system is responsible for the way we see and reach to our surroundings. Now, what will happen if there is any defect on visual field, which is the entire area that can be seen when the eyes are focused on a single point? First, the lesion that interrupts one optic nerve causes blindness in that eye. Then the lesion of optic chiasm causes loss of vision in the different side of the visual field in both the eyes, which is called heteronymous hemianopia. Here, the terminology heteronymous means the opposite side of the visual field and hemianopia means the half blindness. Next, the lesion of optic tract causes loss of vision in the same side of the visual field in both the eyes, which is called homonymous hemianopia. While lesion from the occipital cortex causes less of vision in the same side of visual cortex. In the both eyes pairing the fibers from macula. Moving to the clinical perspective of common diseases of visual dysfunction, which we need to know are hyperopia or farsightedness is a difficulty with near vision, but far objects can be seen easily. The eyeball is too short and light rays come to a focus behind the retina. Myopia or nearsightedness, where far objects appear blurred but near objects are seen clearly. The eyeball is too long and light rays come to a focus in front of retina. Astigmatism is distorted vision because of the curvature of the cornea is not uniform. The light rays do not all come to a single focal point on the retina. Presbyopia is a loss of accommodation mainly because of hardness of the lens. Glaucoma is a degenerative disease in which there is a loss of retinal ganglia cells occurs, primarily caused by increased intracellular pressure. Night blindness is the inability to see well at night or in poor light due to inadequate intake of vitamin A. Color blindness is the inability to distinguish certain colors. Cataracts is the clouding of lens resulting in blurred images. Age-related macular degeneration is a deterioration of central visual acuity because of the hardening of the arteries that nourish the retina. On the research prospect, the cutting-edge research on vision is revolving around the mechanism of color detection. Color vision is an important sensory capability that enhances the detection of contrast in the retinal images. In humans, the perception of color is also described by the attributes color hue, saturation, brightness, and it can be associated with additional sensations, emotions, or call-up memories. So, the research of Scanatman et al. In 2020 provides the conceptually new insight into the neuronal basis of color vision in Drosophila and important visual phenomena like spatial and temporal color contrast and color constancy. This would be helpful in human in explaining the importance of color for us. These are the reference books from which I have taken the materials for the presentation. Thank you for your patient listening. If you have any queries, please mail me.